Okay, let's finish this chapter up. Finally, gee whiz. Okay, so this last section is all on vision. So vision is detecting our light stimulus and then eventually transferring it into action potential so our brain can actually determine what we're actually seeing. So with vision, it's a perception of the light, either that they emit. So for example, this computer screen is emitting light. There is an internal light that is being projected outward, or something that is reflecting light. So such as this heart model, light is bouncing off the surface of it and then just reflecting the light that hit the surface of that object. So just like sound, light is also on a wave spectrum. If something is shorter in wavelength, so how spaced out between your waves, something that's shorter is going to have an increase in energy, and from what we can see, is going to be more of, like, for example, your purple and blue colors versus lower energy waves, so a bit more space in between. That is going to have lower energy, and that's kind of like your red, oranges in that category. And again, stimulation by how much, kind of like we had with the other one where increase in stimulation, so a brighter light is going to have more action potentials and tell the brain, hey, that's a lot of light. Okay, so with our light, it's on a electromagnetic radiation spectrum. So what we can see, this tiny little sliver, is our visible spectrum. There's other light, let's see how to say this. There's other energy wave spectrums of stuff that we can't see. So for example, gamma rays, which is your space radiation, a ton of energy. That's why it can just blast through stuff. They actually have to deal with this in just doing with the equipment, for example, on the space station or on a rocket. Because that space radiation is going to blow through, just go straight through the shielding for the space station or the rocket and possibly affect your equipment. So not just the effect on the astronaut, even the equipment got to make sure it protect it. This one's X-rays for example, like your detecting bone scans, that kind of stuff. So this is why the technician goes behind the wall while you're wearing a lead vest or whatever, because they're going to get exposed all the time. So that's why they just completely go out the room while you're still stuck in there. Ultraviolet, so it's our sunlight, so still kind of possibly damaging light. Infrared, that's our heat. And then our radio and TV waves are a lot larger, so longer wavelength. And this allows them to have a long broadcast distance. So for example, AM has a longer broadcast than FM. This allows it for a bigger, wider spread. So it has a lower energy. That's why some stations that don't have as much money are going to be on AM channel, because they don't have to use as much energy to broadcast than a FM station. Okay, let's get off of that. So with, again, our different spectrum of light, again, UV on one end. So for example, our sun, too much energy can actually cause damage. The light actually is able to cause a photochemical reaction to be detected, and we're able to use that information for the brain, tell us what we're actually looking at. And then on the opposite side, infrared radiation, which we do actually have the way to convert that heat energy signal into something we can actually see. So we're looking at this little dog, whatever kind of dog it is, I don't care. But the areas where his little tips of his ears, or her ears, whatever, eyes and mouth, or little gaps in the fur, that's going to be warmer because it has body heat coming from it, versus the areas that are more covered in fur are going to be a little cooler in color. So there is a way to detect different levels of heat. So actually now, of course, hopefully, cross fingers, that eventually we won't have to be detecting our body temperature, our like, a temperature as we're getting checked. Like right now we're dealing with the whole COVID situation. So the little gun that they're pointing at your head to get your temperature, that's what it's doing. It's detecting the level of infrared radiation so that it can actually detect your temperature. Okay. So let's look at our eye parts. a lot of this I don't care about, so that's why I'm only covering some of it. So eventually in the lab, just in case we accidentally have to do this early, the white part of your eyeball is called your sclera, so it's very hard, tough exterior portion. The iris is your colored portion of your eyeball, and then the pupil is this open space that allows light to go into your eye. Now, the reason why you don't see the light that would be inside your eyeball is because all of it gets absorbed, so that's why it looks dark. So anything that's white is emitting or reflecting all the light back, versus something that is black is actually absorbing all the light. Other pieces of the eye, so we have the cornea, which is on the front surface. That's a very hard, also exterior portion, just to keep you from scratching your eyeball. This little section here is your conjunctiva, so if someone gets conjunctivitis or pink eye, that infection is actually just right there inside the eyelid. Other structures for our eye. So our eyeballs are wet. They're wet because of tear glands. So even though you're not actively tearing up, you're still having a wash of the eye, and that just keeps it clean. And that is using your lacrimal gland producing your tears, and there's different types of tears that we actually produce. That washes over your eyeball, and then it collects into the lacrimal sac, which is behind that lacrimal bone in your eye socket, and then eventually that drains out into your nose. Most of the time, that tears, because you're breathing through your nose, is going to dry out. Or if you are really crying a lot, and a lot of your tears, instead of spilling out over your eyeball, are going into that lacrimal sac, it's also going to come out your nose. So that's that kind of really ugly crying when you just got snot going everywhere. Yeah, that's that kind of, that's what that is. A lot of that's just tears. So same thing with little kids that are having a complete temper tantrum. It's the tears that are collecting into that sac and draining out their nose. So at least you're clearing out the snot. Okay. Eyeball parts. So some of this you actually do label on the test. It must be on the study guide. I need to make sure this is labeled on the slide, though. Okay, so we talked about our sclera, which is on the outside, our hard white exterior. Then on the inside, we have our choroid. So this really dark section here, that's going to absorb the excess light. The retina, which is where our photoreceptors are, so that is the yellow portion there. Our optic disc, which is our blind spot. So in this section, this is where all your axons are coming in from your photoreceptors, so there's no photoreceptors in that spot. So this is where your brain just makes up stuff. So all of our axons come together and collect as our optic nerve, so we can send the information to our eyeball. Other parts in the front. So we have our pupil that allows light to come in, so we change the size of it to determine how much light can come in. Around that is the iris, so there's some muscle there that helps to change the size of the eye. There's the cornea again. So again, our front section of the eyeball. So protect the front of our eyeball from getting scratched. And then as this light comes in through our pupil, it passes through the lens. So that's going to help us focus on what we're looking at. So whether or not we're looking at something that is close by or far away, that lens is actually going to be able to change shape and allow us to focus on the specific light that's coming in. So when we do our eyeball dissection in lab, it's a cow eye, and when you open it up, you'll actually see a little like hard marble, and that's hard because it's been preserved, so it should be softer than that. It feels like a non-preserved eyeball. But that little marble that's on the inside, that is actually your lens for that eyeball. And then the rest of this is called the vitreous body. It used to be called the vitreous humor, so it's a fluid, jelly-like substance on the inside of your eyeball. So when you do that, depending on what happened in the preservation process, you might have a little bit of that jelly left over, or it might all got dissolved away, so it just depends. Something else I was going to say. Oh, another thing. With the retina, so our little yellow layer here, that can actually detect and detach, and most likely it will when you cut the eyeball open. But you'll still be able to see it. It's kind of beige in color, and you'll see it kind of clumped up at the end and still attached at this blind spot. So where the optic nerve is coming out. So if you ever had someone who you heard about their retina detaching. Yeah, they had some major trauma to their eyeball, and so therefore the retina detached. Okay, so how do we actually detect light? So part of our eyeball is actually clear or transparent, and these are called our optical components. So these allow the focus of our light onto our retina. So going from our pupil and into the back of the eyeball towards the retina. So that is the cornea, aqueous humor, lens, and the vitreous body are also called vitreous humor. So again, your cornea is your outside cover, very hard. Your aqueous humor is a little bit of fluid that's in between your cornea and your lens. Then you have the lens, again, that's going to change shape to help focus your light. And then your vitreous body, so once you have focused the light, the vitreous body is going to, your light's going to have to pass through it to get to your retina. So for some people, it could be a pupil thing, but also could be a lens not able to focus thing, and that's why they have to wear glasses. Okay, so when our light gets all the way to our retina, that's where we're going to convert our light energy into action potential. So we're going to take our light energy and convert it using the photoreceptors that are in that layer. We also have a layer of pigment behind there to absorb light, so we're not having stray light bouncing off of something and then going back to our photoreceptor cells and re-stimulating it. So making sure we have a nice, clear picture. Other cells that are necessary in that area, but these are the more important ones. We have our bipolar cells and our ganglion cells. Those are also going to be just part of the pathway to get to the brain. So with our photoreceptors, we have two different types. We have our rod cells and our cone cells, so shaped because of how they look. So our rod cells are called for our night vision or monochromatic vision, so part of our scopic light system. Our cone cells, cone-shaped, are for day vision. So this is for our color vision. So we have three different types of this, and that's how we're able to see in different colors based on which part gets stimulated. So one way of remembering that the cone cells are for our day vision or also our color vision is thinking of, if you have like your rays coming off the sun, the cone cell fits in that little triangle portion of your light ray or your sun rays. So with our rod and cone cells, they actually are packed up against each other, so they're spread across the entire retina. So our rod cells are the taller ones, and then our cone cells are the one at the bottom. So they're all next to each other, not in like specific spaces. So here is our layers at the back of the eyeball. And depending on the damage that might happen or change that might happen, you actually could have changes in how thick some of those layers are. Translation, I had to actually do this and measure all this stuff for a project. Anyway, so here down here is our vitreous body. Light's going to pass through. It's going to pass through our ganglion cells, pass through our bipolar cells, pass through our rod and cone cells, and that's actually where you're going to get to our light detection in closer to our pigment epithelial layer. And then anything else gets absorbed by the choroid in the back of the eyeball. And then this is just the sclera, that white portion, hard exterior portion of the eye. So looking at our different dots, we see that we have way more photoreceptor cells than we have our bipolar and ganglion cells. Because you're going to have in our, for example, in our scopic system, so for our rod cells, you're going to have a bunch of those synapsing onto or converging onto one bipolar cell, and then you have a bunch of these bipolar cells that are going to synapse or converge onto one ganglion cell. Okay, so how does this work? So same ordering that we just saw. Light's going to pass by the ganglion cells because they don't detect light, so they don't care. Pass by the bipolar cells, again, they don't detect light. Go all the way to the back where the pigment epithelium is, and that's where we also have our rod and cone cells, and that's where we actually get our light detection. Then these photoreceptors are going to convert that light energy into action potentials and then send them to the bipolar cells, and then send them to then the bipolar cell sends that information to the ganglion cells, and that axons are creating your optic nerve to go to your brain. So the neurons are going to converge onto one neuron. So you're going to have information coming from multiple cells onto one neuron. So getting that information in. So we don't have, even though we have pretty good detection of details, it's not like, you know, super much. So that's why some of that television stuff, it gets to the point of, yeah, this doesn't really actually do any better. Like, for example, the new one, when we get to the color section, there's one that actually has a yellow as a color, and it's like, but we don't have a yellow photoreceptor. So what's the point? Okay, so let's look at generating a visual signal. So again, we actually have to have light to actually activate our photoreceptors. So the ones up top. And so for this one, we're looking at our rod cells. So inside our photoreceptors, there's going to be a photochemical reaction. So there are pigments in them that are able to react to different types of light. So when there is no light being detected, the photoreceptor cell is actually going to inhibit the bipolar cell from actually generating an action potential. So there's not going to be any signal being sent to our ganglion cell. However, when you actually get your photoreceptor, get light, and so therefore the photoreceptor detects the light, then your rod cell is going to no longer inhibit your bipolar cell from sending an action potential. So now that our bipolar cells are able to send action potentials, they're able to stimulate the ganglion cells. Those ganglion cells are going to make action potentials that tell the brain, hey, we're looking at stuff. So with our two systems, the number of bipolar and ganglion cells that are synapsed onto is determined by the type of photoreceptor. So for our rod cells, multiple rod cells are going to synapse onto a ganglion cell, and then, are going to synapse onto a multiple rod cells are going to synapse onto one bipolar cell. One bipolar cell is going to synapse onto one ganglion cell, and then all of that information is going to be sent onto our, onto the optic nerve into the brain. So with our rod cells, they are more sensitive to light. So this is why they're more part of your night vision. So collecting as much light as possible and pulling in all of that information, all the little bits of light that we can get, and then sending all of that onto one neuron. So this gives us a coarse image. So just giving more of a general type of image. So like, for example, at nighttime, where everything kind of looks like this bluish purple color because that's the pigment color that's in those rod cells. But also you don't have much detail on what you're seeing. You're kind of seeing kind of more general shadows. And that's why certain stuff can look really funny at night, is because you're kind of getting a very generic coarse image. For our photoptic system, we're using our cone cells. These are less sensitive light because you have a little bit of light here, a little bit here, a little bit here, a little bit here. So all these little tiny pieces of light are all individually being sent to the brain. And so you don't get this collective power of light as you would with our scotoptic system. So with our photoptic system, you have no convergence on our, for multiple photoreceptors onto a bipolar cell. Here, it's a one-to-one situation. One cone cell to one bipolar cell to one ganglion cell. And then each of these ganglion cells is sending that tiny little bit of information to the brain. What's good about this is that we get a fine, detailed image. So more definition on what we're seeing because we're seeing differences between the little bits and pieces of information. Good Lord, it's 19 minutes. Okay, so with our being able to see stuff in color. Again, our rod cells uses rhodopsin, which is a purplish color. That's why stuff at night kind of has that kind of coloring. It's going to have a wavelength detection, mostly at 500 nanometers, which is in that spectrum there. So this black line that's showing kind of what it can detect. because it only detects has one type of pigment, it only can detect kind of like one generic information coloring. So it can't distinguish one color from another. However, for our cone cells, we have 3 different kinds of cone cells. And the reason why we have 3 different kinds is because they have 3 different pigments. So we have a blue one, a green one, and a red one. So we have S, M, and L. So with our 3 different colors, they cover the entire spectrum. So for example, even though we don't have a yellow one, so our red, green, blue, those are kind of like our primary colors. So for example, yellow is going to be a mixture between stimulating both our M and L cones, which is our green and red. Now here's a problem. If you lack one of those cells, either the M ones or the L ones, so either the green ones or the red ones, if you're looking at these 2 lines, they look exactly the same color to you. Why? Because there's so much freaking overlap between the 2. So for people who are missing one of those, that leads to color blindness. And this is why you get the cool little sunglasses now. And you actually can split that up and can see a difference between the 2. So for example, if someone is colorblind and they're looking at this circular structure here, someone who is not colorblind sees the number 74 with using the blue and green dots that are embedded in that picture. However, someone who is colorblind will only see 71 in the picture because they can't tell the difference on this corner of our 4. So we have our 7, and then this corner of the 4 and this little edge piece of the 4. Because the green and the red look exactly the same, then for them, they only can detect the 7 and the 1. This is also why our stoplights are arranged where they do. So you have a difference between the bottom green color and the top red color. So people who are colorblind know exactly, should I be going or should I be stopping? So they used to actually be sideways, but then because of the colorblind issue, they flipped them. So red's on top, green's on bottom. Okay, last piece on our vision, really long video, sorry about that, is determining being able to see in 3D. So this is called stereoscopic vision. So again, we're taking in information from both eyeballs and sending it to the brain and kind of comparing what we see. And this allows us to see in depth perception. So the overlapping visual fields, again, we got information coming from 2 sides. Our eyeballs are in the front, like for example, predator animals, the example given there as like a owl. What are you doing? Stop that. There we go. For them, they can see in the overlap, able to see something in 3D. So seeing, like, for example, what I'm looking at right now, I see my cell phone. I can tell that it's in front of my computer screen because I'm comparing the information from both sides of my eyeball, and I can see a little bit on one side of my cell phone, a little bit on the other side, and I can tell, oh, my cell phone must be in front because I'm missing some of the information from my computer screen on one eye, but it's not missing in the other eyeball. So comparing the 2 sides, I can tell that my cell phone is closer to my face than my computer screen. So this allows us to see in 3D. Unfortunately, it also leads us to have a blind spot behind our head, unless you're a mother. Okay, just kidding. But blind spot behind the head because we just don't have a visual field detecting stuff back there. For the other type of vision, that's panoramic vision. This is where the eyeballs are on the sides of the head, so very little or no overlap, and so you don't have depth perception. So they kind of see stuff in general, kind of like how if I just start looking at something on the sides of my eyes, so this would be like in your peripheral vision, that's what they see all the time. So they just kind of get generic information as in, is something about to kill me? Is something about to eat me? Is something about to kill me? Something about to eat me? That's why they kind of look kind of weird and freaky, because they're always kind of like, what is that shadow? What is that shadow? Is that about to get me? It's because they just can't see stuff that clearly because they only see in 2D. So if you ever looked at, and this is your prey animals, so if you ever saw a bird and it kind of does this bobby head motion thing, it's because it's sticking its head out to see a little bit further back and then comparing to what it sees back here. So it kind of goes back and forth, and that's kind of how it kind of makes up for not being able to see in 3D. Now, don't ask me what happens when they're flying at high speed and it's like, you gonna run into something? That's probably partially why they do. Okay, so that is the end bit of chapter 16. Again, fill out the study guide. You should figure that out now. And if you have questions, let me know. And we're done, at least for AP1. See ya!